Hello everyone, I am Dr. Dipti Dhere and today I am going to discuss about active site and mechanism of enzyme action. In last lecture, we have discussed the basic concept about the enzymes, which includes the term enzyme was firstly coined by the William Punney. The enzyme means in yeast. The enzymes are called as a biocatalyst because they are active in the living system, which is also called as a biological system. They are called as a biocatalyst but not the catalyst. The enzymes are biocatalyst which increases the rate of biological reactions by decreasing the activation energy. Then activation energy, it can be defined as the minimum amount of extra energy which is required by the reacting molecule to get converted into the product. As enzymes are protein, they are synthesized by the ribosomes on the endoplasmic reticulum in case of the eukaryotes. While in case of the prokaryotes, such cellular organelles are not present. And that's why the enzyme is synthesized in the cytoplasm by the ribosomes. Enzyme, they are very specific for their action as well as their substrate. Ribozymes are the enzymes which are RNA in nature. While most of the proteins, most of the enzymes, they are protein in nature. Then we will discuss in today's lecture about the enzyme, particularly the active site of enzyme and the mechanism of enzyme action, where we can define the enzyme as the biocatalyst which increases the rate of biological reaction by decreasing the activation energy and without being consumed and remain unaltered at the end of reaction. Where the biochemical reaction are the transformation of one molecule into another, which are mediated by the catalyst called as an enzyme. Uncatalyzed reaction, they need lots of time. And that's why the enzyme, they play an important role in maintaining this living system. Then it is said that all enzymes are protein, but all proteins are not enzymes. Then why only some proteins are called as an enzyme? What makes it special to say as an enzyme? What makes enzyme different from other protein? So answer for all the questions is the active site. The active site, it is a part on a protein which make it a special so that it can catalyze the reaction and hence it is called as an enzyme. Then what is active site? The active site is the region on the enzyme where substrate molecule binds and undergo biochemical transformations. And these biochemical transformations are also called as the biochemical reactions. Then what is substrate? The substrate is nothing but the reactant in biochemical reactions. The substrate, it reacts with the enzyme to get converted into the product. When enzyme binds to the substrate, it forms the enzyme substrate complex. And during enzyme substrate complex formation, some necessary interaction occurs between the enzyme and substrate so that substrate is get converted into the product. In the given diagram, the reaction of enzyme is shown with the substrate, where E stands for the enzyme and S stands for the substrate. When enzyme reacts with the substrate, it forms the enzyme substrate complex. At the enzyme substrate complex, all necessary interaction occurs between the enzyme and substrate, and so the enzyme get converted into the product. And here the conformation or structural configuration of that product, it is no more compatible with the enzyme or the active site of the enzyme. And here the enzyme kicks off the product out of its active site. And here enzyme is again free, unaltered, unconsumed, and again it is ready to react with the similar substrate. Then about active site. What is active site? The active site of enzyme is the region. It is a micro environment on the protein that binds the substrate. It binds the substrate as well as it can bind with the cofactor and prosthetic groups. It contains the chemical groups that help to hold the substrate. Active site occupy more than 5% of the total surface area of the enzyme. Active site has specific shape due to tertiary structure of protein. When protein is folded into its secondary structure and tertiary structure, at that time the interaction between the amino acid in the polypeptide takes place in such a way that all reactive amino acids they come together to form a particular microenvironment on the protein, and that reactive microenvironment on the protein is called as an active site. The change in shape of protein affects the shape of active site, because if the shape of protein is changed, at that time the structural conformation of active site it is changed. The reactive amino acids which are creating a special environment for getting reacted with the substrate, it get disturbed, and that's why it affects the shape of active site, and ultimately it affects the function of the enzyme. Then active site, the total area of the enzyme, it can be categorized as part other than the active site and the particular active site. Here the active site again can be categorized as binding site and the catalytic site. Active site has some amino acid residues which can bind with the chemical groups of the substrate, and these chemical groups they come under the binding site. Then what is catalytic site? The amino acids which are present in active site which react with the chemical groups present in the substrate, they are included in the catalytic site. And binding site and catalytic site together it gives the active site. Or active site, it is made up of binding site and the catalytic site. Then what are the properties of active site? The properties of active site, they are dependent on the size and shape of the active site, polarity and non-polarity of amino acids, then positive and negative charges of the amino acids, then hydrophobic and hydrophilic nature of the amino acids, and special properties of the cofactor. Here the size and shape of active site, it can be created specifically to fit around the substrate. Particular protein, it is folded in such a way that it can bind with a particular type of substrate, and hence here the enzymes are very specific for their substrate. The microenvironment or the active site, it is formed in such a way that it can bind with only a particular type of a substrate, and hence the size and shape of active site it determine its activity with the substrate. And if the shape of active site it is disturbed, then enzyme it remain no more active. Then what is polarity or non-polarity? In active site, some amino acids are polar in nature and some amino acids are non-polar in nature. Polar amino acids they interact with the polar chemical groups on the substrate, while non-polar amino acids they react with the non-polar chemical groups of the substrate. And here the active site leads to better fit with the substrate. Then positive and negative charges. Positive charge on amino acid attracts the negatively charged part of the substrate, and negatively charged part on active site it attracts the positively charged chemical group on the substrate, and this again leads to proper fit of substrate with the active site. Then hydrophobic and hydrophilic nature. Here the phenomena that is like attracts like is followed, that hydrophilic amino acid they interact with the hydrophilic chemical groups of the substrate, and hydrophobic amino acid in the active site, they interact with the hydrophobic chemical groups of the substrate. And here again it improves the interaction of the substrate with the active site. Then next character is about cofactor. Some enzyme they need cofactor for their activity, because here the active site it cannot directly interact with the substrate, and cofactor here act like a mediator, or it act like a adapter molecule which interact both with the active site as well as with the substrate. And here, due to proper interaction of the cofactor with the substrate as well as with the enzyme, the reaction occurs. Then about active site. Enzyme has distinct cavity or cleft to which substrate bounds. This cleft is called as the active site or the active center. In active site, amino acids are grouped together in such a way that their arrangement enables them to combine with substrate. All earlier said interaction make the active site special, and due to all these characters, such an environment is created in the active site which make it possible to combine with the substrate. The active site has reactive amino acids which may lie widely separated in polypeptide. In polypeptide, different amino acids they may be separately present or distantly present. But when polypeptide is folded due to the charge interaction or the interaction which include hydrophobicity or polar nature, the folding occurs in such a way that all interactive or reactive amino acids they come together. They may be widely present in the polypeptide or distantly present in polypeptide, but in active site they come together and interact with the substrate. Here in this diagram it is shown that polypeptide it is the primary structure of protein, and this polypeptide it is folded into alpha helices and the beta sheets, which is the secondary structure of protein. And alpha helices and beta sheet they again interact to give the tertiary structure of protein. And in tertiary structure of protein, a folding occurs in such a way that the interactive environment is produced in the protein, and that interactive environment is called as the active site. In some cases or in some proteins, more than one polypeptides are present, and in only those protein quaternary structure can be seen. The folding of polypeptide occurs in such a manner that distantly placed reactive amino acids they come together to form an active site. The substrate molecule bind to the active site in such a way that it causes distortion of chemical bonds in substrate. This increases the reactivity of substrate with the enzyme and speed up the reaction. Here in this diagram you can see the tertiary structure of the protein, where you can see that differently situated amino acid or widely separated amino acid they come together and interact, and due to their interaction a proper folding takes place. Here you can see the hydrophobic type of interaction, then ionic interaction, disulfide bonds and hydrogen bondings are also seen. So all different types of interaction occurs which are due to the different properties of the amino acid present in that particular polypeptide. So it is not necessary that all protein they fold in the same way. The folding of a particular protein and formation of active site and the structure of active site it depends on the type of amino acid present in the polypeptide, and it decides the structure of the active site. Then mechanism of enzyme action. The total mechanism of enzyme action it can be classified into two types, or two types of interactions occur between the enzyme and the substrate, which include the thermodynamic changes, which are the energy related changes during the reaction, and some processes which are actually present at the active site. Then what are the thermodynamic changes? All chemical reactions have the energy barrier, and that energy barrier it is called as a activational barrier. And for the uncatalyzed reaction, this activational barrier it is high, and that's why uncatalyzed reaction they need more time to complete. On the other hand, enzyme catalyzed reaction it has very less activational barrier, and that's why the enzyme catalyzed reaction it can smoothly run and it can be completed within a fraction. Only few products or few substrate can cross the activational barrier and can get changed into the product. That is the main reason due to which rate of uncatalyzed reaction is slow. Enzyme provides an alternative path for conversion of substrate into the product, where enzyme holds the protein, it holds the substrate in such a way that the interaction between the substrate and enzyme it happens, and substrate can get converted into the product. The enzyme forms the transitional state which has low activational energy, and hence it accelerates the rate of reaction. Hence the rate of reaction increases manifold in presence of enzyme. And here the total energy of the system remains the same, and the equilibrium state is not disturbed. So in this diagram you can see that a man is pushing ball to the other side of mountain. Due to high height of mountain, the man has to invest lots of energy to push the ball to the other side of mountain. The height of mountain it can be considered as a activational barrier, which is same as in the uncatalyzed reaction. While in enzyme catalyzed reaction, this activational barrier it is very less, and that's why you can see that in second diagram the man it can roll the ball to the other side of mountain very smoothly. And here the enzyme play an important role. It holds the substrate in such a way that the transition state it is reached fast, and hence the enzyme catalyzed reaction it occurs in very less time. Then processes at the active site. What actual processes occurs at the active site? These includes covalent catalysis, catalysis by strain, catalysis by proximity, and acid-base catalysis. These are the four mechanisms by which substrate and the enzyme it occurs. Covalent catalysis, catalysis by strain, catalysis by proximity, and acid-base catalysis. These four interactions they may occur together or in groups, or any random combination of these catalysis can be used by the enzyme to interact with the substrate. That depend on the type of enzyme and the type of substrate. So overall function of enzyme can be summarized in this diagram. So in first case here we can see that enzyme is free which interact with the substrate. There is a formation of enzyme substrate complex, and at the enzyme substrate complex necessary interactions or positive interaction occurs between the active site of enzyme and the substrate, and as a result of these interaction the substrate get converted into the product, and as product it is no more complementary or compatible with the active site, it is kicked off by the enzyme out of the active site. And here the enzyme is active again to interact with the similar substrate. Here the enzyme is unaltered and unconsumed. And that's why the enzyme, they are required in very less quantity during any biochemical reaction. So that's all. Thank you.